Good day, this is Professor Will, CBMD PhD. Today's Saturday, July 24th, 2021. It is 4:48 and 27 seconds, AM Eastern Standard Time. Today's research, organ rejection. So let's hop to it here. Missing self contributes to organ rejection after transplantation. Study identifies a major role for immune cells, called natural killer cells, in organ rejection. Date July 22nd, 2021. Source, American Society of Nephrology. After kidney transplantation, natural killer cells of the recipient become active because they miss cell proteins on donor cells. These cells contribute to organ rejection, in addition to traditional modes of rejection involving T cells and antibodies. Immune cells called natural killer cells contribute to organ rejection after transplantation because they miss cell proteins on donor cells. A better understanding of this process may help clinicians prevent and treat organ rejection. And transplanted organs are recognized by the immune system of the recipient as foreign or non-self, which leads to rejection of the organs. Rejection is prevented or treated with drugs to suppress the immune system, mostly targeting T immune cells. However, rejection can still occur despite such treatment, not only because T cells may not be completely suppressed by the therapy, but also because of antibodies and natural killer cells that target the donor tissue. Natural killer cells play an important role in the human immune system, as they are involved in recognizing and killing harmful cells, such as tumor cells. And these harmful cells sometimes attempt to escape immune detection by decreasing MHC proteins, which are proteins expressed on cells that allow T cells to bind to, recognize, and tolerate itself. And this mechanism renders the harmful cells invisible to T cells, but not to natural killer cells. And through their KIR receptors, natural killer cells can detect the absence of these MHC proteins, and therefore kill the harmful cells. And this constitutes a very important defense mechanism. In transplantation, the donor cells and the transplanted organs are not escaping immune detection by decreasing MHC expression, but these donor cells express different MHC proteins than the recipient. The natural killer cells of the recipient therefore miss the self-MHC on these donor cells and become active. And this is exactly what they found in their study of 924 kidney transplantations, that the missing cell predicted by genetic analysis of the MHC molecules of donors and recipients, and the genetically determined KIR repertoire of the recipients, is predictive of rejection in kidney transplanted biopsies. And therefore the research study shows that genotyping the donors and recipients, not only for MHC, as is done in routine clinical practice, but also for KIR, will enable us to assess the presence or absence of missing cells, and improve the risk assessment of kidney transplant rejection. Furthermore, the research findings demonstrate the importance of these natural killer cells after transplantation and suggest new ways to prevent or treat kidney transplant rejection. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.